to Rwanda, because Rwanda has, thousands of hills. That's what I was told when I was in Rwanda. People have generally destroyed the forest in the flatter areas, but then so the forest is now becoming narrower as you go up higher onto the mountains and onto the hills, and then these areas are increasingly becoming, endangered. Because of disturbances like wildfires, like farming, illegal logging, we can mention but a few. So these are contributing to the distraction of these areas, coupled with climate issues, the El Ninos. We'll talk a little bit about that in a few. So these are, different disturbances that is affecting montane forests in our current dispensation. So because of this, there's been global consensus and to with global targets and objectives to restore degraded forest. Um, as we all know, by 2030 to restore a huge amount of degraded forest. I do not know how that target is going to go, but certainly Africa has contributed a lot. But one thing that strikes me is that for us to contribute to restoring degraded forests, to meet these ambitious targets, will be to have a scalable restoration strategy to get there. And so many people have done a lot of research, and they have proven that some restoration strategies are more scalable compared to others. And today we'll talk about assisted natural regeneration as one of the restoration strategies. Usually people will go for plantation. I'm not pro-bias of plantation. It is great. It is very fast. It also provides timber and then supports, carbon sequestration at the end of the day. But if we want to restore biodiversity, if we want to restore the soil, the landscape in itself, in its entirety, we would want something that is more interesting. And then I would always say that natural regeneration is the best option to go. But usually it takes a lot of time. So what people would do now is to assist the system. The natural regeneration would more like leaving the system by itself, for the forest to regenerate by itself. Species, early successional species come in, and then they grow very fast. Those are pioneer species, and then late successional comes in later. They go through stem exclusion phase. Like the forest, moves into different phases until we get the natural forest back. But that process takes a lot of time. So because of that, now people like to assist the natural system. And that is what we classify as assisted natural regeneration. It's, basically removing perturbation, disturbances, any form of disturbances, if it is grazing, wildfires, illegal logging, any form of, illegalities or disturbances restrain that can restrain the natural processes. Once you remove that, ensure protection. And then in areas where you have arrested succession, I'm trying not to use the terms. In areas where the system is very difficult to regenerate by itself, you would want to go in and then apply some strategies to enhance the system to regenerate by itself. So this is what we're going to talk about today. Um, we had similar areas in Nyungwe National Park that has undergone wildfires, and so a little bit background to what we're discussing today. In 1997, um, the forest before 1997, the Nyungwe National Park has always been pristine, right? As we all know, because we're familiar with this forest. has always been pristine, a lot of interesting wildlife. I saw chimps in there because I did some hiking in there. And I had to run. And that was very interesting for me because I had to run and chase these chimps because I just wanted to take a picture and they would not stop. So I had to follow them for like 1.5 hours before I could get a perfect picture. And that was interesting. So just running over this. So, and we have the black and white Columbus monkey. So these are things that I have seen myself, and there are tons of biodiversity from wildlife to plant species in this particular area. It has always been pristine, very interesting forest, very misty in the morning. So it's very beautiful scene. But then, somewhere in 1997, during the El Nino years, coupled with other anthropogenic activities like honey harvesting, people had beehives and then they used fire, and then it was also extremely a hot period during the El Nino time. So, coupled with the climate and anthropogenic activities, these areas were set into fires. So fire affected this particular forest in 1987. And then it covered about 12% of the forest, which was a lot in my thinking, for a place as pristine as this. And then I think that even currently, some of the areas that were affected by the wildfire still remain undeveloped in terms of structural development, in terms of forest. Now, these areas that were affected by wildfires were later colonized by Pteridium aquilinum. As you can see, the picture, you can see that I'm pointing with my black arrow pointing towards as Pteridium aquilinum. I'm sure many of us are aware of this particular species, which is also called the bracken fern, commonly called the bracken fern. So these species colonized. Can you hear me?
Yes,
Yes, can hear
but
you.
We have
Okay.
a background noise from Twahirwa, and I think he can mute himself.
Okay. So
Twahirwa,
these
kindly mute. Yeah, you can go on.
Thank you. So these species colonized these areas that were affected by wildfires. And then in 2003, the management group of this particular park, together with WCS and specific people like Felix Linda Happy, um, I believe he's still working with the African parks. I hope he's there, and Anna Mosera and Amy. So these people, coupled with the management, came together and decided to figure out ways and means to remove these particular fern in a way that will not a time or a period that is manageable enough, for scaling. Because obviously, when you have people going in there to clear these areas, it also costs some money. So you don't want to do it all the time. So it has to be in a specific period. So they had 2 months, 3 months, and 5 months. And then they came to the conclusion that the 3 month period is the most effective way of restoring indigenous species. And then also it was very cost effective. The 5 months was just way too much. 2 months was just way too short a period. And then, because it would cost a lot of money to clear the fern in every 2 months. So 3 months was very optimal. And at the same time, they realized that, the removal of these fern um, supported about 20 indigenous species in the treated site. So the treated site where fern was removed every 3 months consistently for 3 years is what we would term as the assisted natural regeneration site. Hereafter, I will be calling it ANR site. All right. But this was years ago. We do not know, well, now we do, but we didn't know before now, we didn't know how these treatments applied has performed over the years. So our research went into it to see the 20 years post-treatment long-term recovery trajectory of these specific areas that were affected by wildfire. Now, they established new set of ANR plots, assisted natural regeneration sites. There were about 4 of them: Mogoti, Gasera, Kitabi, and Mubuga. But our research narrowed down to 2 ANR sites, which is Kitabi and Mubuga. We selected our plots from these areas. These maps are from Felix, from the report that was shared with me. And so what was our objective? We wanted to assess the variation in forest structure across unburnt forests, the assisted natural regeneration site, and then the unassisted natural regeneration site. And then again, we also wanted to compare species richness and diversity across the natural forest, the assisted natural regeneration, and then again, the areas that were not treated. So they had areas that were treated and areas that were not treated. And then also we wanted to evaluate differences in species composition. Now we'll go back to this map. So you can see that we have this yellow, and I don't know if it's magenta color in Mubuga and Kitabi. So those polygons are the treated areas. The area of these plots ranges from 250 meters square to 750, right? And then in between, some of the in between these areas are the fern or bracken fern lands where no treatment, there was no weeding there. There was no released weeding, I would say, because what they did was that they removed the bracken fern and allow every form of indigenous species to survive. They maintained those ones and then remove the bracken fern every 3 months consistently for 3 years. So what we did was that we compared the composition, the diversity, and the forest structure of the treated side, untreated side. We compared that to the natural forest, adjacent natural forest in the area. And then we hypothesized that assisted natural regeneration plots will exhibit higher stem density and mean height compared to the fern lands. And again, species diversity will be higher in the treated site compared to the untreated site. And then we were expecting that the highest species diversity will be in the natural forest. And then our last hypothesis was to check to see species composition. And then we were expecting that species composition in ANR will be more similar to the natural forest compared to the untreated side. So that's more like a beta diversity. So we did our assessment on stem density, height, basal area. Well, because of time, I didn't include all of that in this particular presentation. But we assessed the height, the stem density, seedling density, basal area, all of that for forest structure. And then we assess richness and diversity as well. And then again, we use an NMDS, generalized linear model as well, and variance partitioning to assess the the composition or the beta diversity between plots. All right, and the all analysis was done in R. So as we are all aware, I'm sure you're very familiar with Nyungwe National Park. But one key thing on this slide is that because the fern lands were just adjacent the untreated site, we paired them up on the same hill, because what we did was that we did not collect data on soils. So because we wanted to account for soil variation, we made sure that the plot selection was very keen. The fern land and the treated site, we made sure they were all on the same hill because we wanted to account for soil variation. So it would not be interesting to collect data from a fern land that is very far away from the treated site. So they were all on the same hill, and that's the reason for the pairing. So as you can see, the yellow and the red, these are treated and untreated sites found on the same hill. And then we compared it to the unburnt area across the street on the other side of the road, because we were assured that these areas has never experienced wildfires, and the fires was on the right side. And so we needed to have a pristine area that has never experienced wildfires for comparison. And so that's what we did. It's basically the same thing. So I was sampling design. We had 15 plots per forest condition, 15 plots in the ANR site, untreated site, and then the natural forest. So in total, we had 45 plots. And then we collected data from tree diameters, ranging from 2 centimeter dBH and above. And then we collected height from these plots. We considered this threshold as the overstory. So in our analysis, we grouped the analysis into two different folds: overstory and then understory. And then we considered understory as seedling count that falls within the category, 20 centimeters and above in terms of height, but also less than 1 centimeter dBH. So if we fall within this threshold, that was considered as a subplot, which is our understory. So we had 4 subplots at the diagonal ends, which was 1 meter by 1 meter. And the main plot where we collected tree diameter greater than 2 centimeters was 10 meter by 10 meters. So it was a 100 meter square plot. Right, now let's go straight into our findings and let's look at the forest structure. We have a lot, as I already mentioned, we have stem density, basal area. So we do have a lot, but then we have heights and stuff, but I would limit my presentation to stem density and height because of time. But if you see the stem density, you can actually imagine what the basal area is going to look like. So let's look at the stem density. Obviously, we had a lot of stems in the assisted natural regeneration area. You can see the picture above, the first picture, in and out stands. So these stands are basically in a stem exclusion phase. And you look at the chart or the graph on the left, you see that ANL was the highest, looking at the letters. So the letters indicate which was the, where the differences were. ANL is not comparable to NF because they have a lot of stems and they are in an early stem exclusion phase. And so, of course, obviously, there are regenerating, they are very young. So they're growing. But then the natural forest is more of, an old forest stand. So we don't expect a lot of regeneration going on. We don't expect a lot of, stem exclusion. That would be more trouble. That would mean that something definitely has happened into that forest. So we're expecting very large trees, but few stems, right? And as you see in the picture below, these are untreated stands. These are natural forest stands. And of course, they are very large trees, but few stems. So because I didn't put basal area here, I'll talk a little bit about it. The basal area of the assisted natural regeneration and the natural forest, the reference forest, were very similar. Not necessarily because the trees in the assisted natural regeneration plots were bigger, but because there were many. There were numerous of them. So putting a lot of stems together that are many can be comparable to few but large stems. So that makes the basal area look more similar to the natural forest, but they're not very similar because they achieved it through different means. One key point is that the untreated area, which is the orange rings, the C, is not comparable to any of them either. These forests remains structurally developed in some sense. So one key thing is that without the intervention, this forest wouldn't have developed at all. And then let's look at the mean height. The ANL site is comparable to the reference forest in terms of height. But again, the key point is that the untreated site remains structurally still different. There aren't a lot of trees as much. Now that I think of it, I wanted to put a picture here. There aren't a lot of trees as much. The entire place is almost all the time, all the plots that we visited, they're covered in bracken fern. So that without any intervention, if we wanted to go the natural way, these areas will still remain undeveloped after nearly 20 years, right? So even after nearly 20 years, that was like about 18 years, we still don't have the untreated side comparable to the natural forest. However, the treated side is very much comparable to the natural forest. And then we wanted to see if our sampling was done correctly and was quite even sufficient. So we use species accumulation curve to do this. And then if we continue to sample in the natural forest, which is the green bar, the unburnt forest, we would still definitely have some more species if we continue to sample. And the same for the assisted natural regeneration side. If we continue to sample, we will still have a few more species, even though it is curving down. But the natural forest keeps going up. And so even if we had continued sampling, we will still find a lot of species in that area. However, if you look at the orange bar, it went up and it became like flat. It's becoming flatter. So if you continue sampling, what it means is that we are not going to find any other new species. It's going to be the same thing we found already. So let's look into what the species richness look like in that area. Now, in terms of richness, again, this proves our hypothesis. Air now is comparable to the reference forest, which was exciting to see that this ANL treatment is actually working and it's giving very interesting results. And now comparing the two to the untreated sites, it's still very different in terms of richness. There are basically no species in that area. Well, there are few, but comparing that to the treated site, they remain, undeveloped in some sense. And then we look at the diversity, our second hypothesis. So we hypothesized that we wanted to have that the ANL treatment will be more similar to the natural forest. And that actually also proves right. But again, in terms of diversity. We looked at it in terms of overstory and understory. Both of them looks very different. The untreated side is not comparable to the treated side, neither is it comparable to the natural forest. Again, in terms of diversity, there aren't as much. Statistically, this is also correct. Even though we know from field observation there weren't much, and then statistically this also proves correct. And now it's the best in terms of restoring species richness and diversity at the same time. But then let's look at what happens in terms of composition. So now we use NMDS, non-dimensional multi-scaling. We use NMDS to check for the composition in the overstory and then also in the understory as well. Now, looking at the overstory, this is very interesting. You see where the circles are, the green and the blue. The blue is the treated side, the ANR side, and the green is the reference forest. The orange, again, is the untreated side where we have a lot of bracken fern. Now, these are not merging. There's no overlap in the overstory. What it means is that our hypothesis was that the ANR site will be very much similar to the natural forest. But in terms of overstory, looking at it, the ANR site is not the same. They are very different. They are not similar. It's not the same as the reference forest. There are no overlaps. If we had the dots, the blue and the red, overlapping each other in the overstory, then we would say that is very similar. But in terms of composition wise, the proportion of species, the type of species that are there and the proportion of those species, they are not similar at all. Yeah, there's a little bit of overlap in the untreated side. Now, looking at the overstory, in general, these are not similar. So we will dive deeper into this, why these are not the same. I mean, we want to know, why is it that we apply the ANR treatment, we remove the bracken fern, allow the species that are there to, survive, and yet still looking at the species in the overstory, they are not the same or comparable to the natural forest. Now, so we switch to look at it in terms of the understory species. What is in the understory species? Are they comparable to the natural forest? And that gave us, a different second. That gave us a different overview. There were a lot of overlap. So as you can see that the charts there, you see the green and the blue mixed up in the center. What it means is that there were a lot of overlap. And what it's telling us in simple terms is that ANR is comparable to the natural forest in terms of understory species composition. The type of species that are there, the proportion of those species are very, very similar to the natural forest. So we dive deep to look into what sort of species are found in this area. What makes it different in the overstory and what makes it similar in the understory? So we use generalized linear model to answer this. Now, in terms of the reference forest, we'll see the proportion of early and late successional species. We have a fair amount of early and late successional species in the reference forest. There are some quite few early successional species in those areas as well. Those basically are pioneers when there's an opening or a gap. These are the species that first jump in and they grow very fast, close the canopy. Once they close the canopy, other species that are not very light demanding would now begin to grow underneath these particular early successional species. And we have a fair proportion of that, not so much, but a very good proportion of that. But it's not so for the ANR site. What is happening is that we have a lot more of early successional species. So when treatment was applied, the ANR was applied in those areas. What happened was that because this was already an open area, the species that first came in were species that really loved the sunlight. So there were species that, grew very fast. There were more of early successional species, sun-loving species like Macaranga kalimanshirika. So they grew very, very fast and they dominated those areas. So we still do have late successional species because they have, the close canopy. But there are very few of that in the treated side. Interestingly, we go into the understory, and the understory, as we saw in the NMDS chart, again, in the generalized linear model, GLM, the ANR is comparable to the reference forest. They are statistically not different. Proportionally, we have fair amount of early and late successional species, almost the same, which is great. In the understory, ANR is really supporting the kind of species that will make the forest in the future look more like the natural forest. One interesting finding is that when we look at the untreated sites, both in the overstory and understory, it almost remains as though it is never changing. It's almost like it's not changing. The same amount of proportion in terms of early and late successional species. So without the intervention, without the ANR treatment, these areas, nearly 20 years after the wildfire, will still remain undeveloped in all directions in terms of diversity, in terms of richness, in terms of structure, in terms of composition. It will still remain the same nearly after 20 years. So this is really going to take a very long time to restore back to its original position. So, most people say that, oh, natural regeneration is the best. There's no doubt about that. I would say that it is the best, but it depends on where we're talking about. It depends on the landscape. It depends on the area of interest. So for me, based on this research, I would not say that natural regeneration is essentially, very useful in regenerating montane, post-fire montane forest. Because now this place is very, very different. The species in those areas are not well adapted to wildfires, as you will have in the savanna areas where species are well adapted and then they can just regenerate by themselves, or even are protected from the fires. It is not so in the montane areas. And if you want to leave those areas to regenerate by itself, that's also going to take a lot of time to achieve. Based on this research, that's not going to happen anytime soon, because this is nearly 20 years. And then we have ascertained that ANL is proving to be very, very effective, in scaling this process. Now, we looked at also variance partitioning. We tried to see if the treatment applied had any influence on the composition. This is basically supporting the NMDS and the GLM. So the variance partitioning is basically we're trying to assess if the treatment applied supported the composition, or it's the differences are due to distance, close to a nearby forest, or perhaps any environmental conditions. In our case, we use aspect and then slope. So is this basically, the regeneration and how effective this has been? Is it because the treated site is close to a nearby forest? Is it because the treatment is actually the one that is working? or it's because of the environmental factors, how slopey the area is, because this is really, really mountainous. So we use variance partitioning to check this. Now, again, in the overstory, you can see that, values that are less than 0 are not shown here. So if it's empty, it means that the value is less than 0. So what this means is that the treatment in the overstory is not the reason for the change we see. However, what is the reason for the change that we see, the compositional change, is because the treated site was close to nearby forest. So that, in terms of that, it is space. That is about 0.0707. So being close to a nearby forest can also enhance ANR. That's what this is basically telling us. Because you have seed sources coming from the close forest, birds moving from the close forest and, dropping the seeds in the treated site. So these basically influence a lot more. There were also a little bit more influence from the environment, but it wasn't as much. So in terms of the overstory, what really influenced the composition was the space, how close that is to the natural forest. But in terms of understory, the story is different again. In terms of understory, it is the treatment that is making the composition look more similar to the natural forest. So this is very interesting, kind of supports both the NMDS and the GLM. Now we'll look into the species that are found in these areas. Again, I already mentioned Macaranga kilimanjarica. Interestingly, this particular species is found across all the treatment site, in the natural forest, in the treatment site, and again, highly, present in the untreated site. About 80% of the species found in the untreated site is Macaranga kilimanjarica. Now, this is not very surprising, but it was a little bit surprising for me in terms of the natural forest. I expect this to be an old forest that is closed up, so you don't really have a lot of species that are, early successional species. That means something probably happened and there were open gaps and allowed these species to grow. But it's not much though. It's about 44%. And then we well expected this to happen. We expect early successional species to take over the assisted natural regeneration because of the open gap, full sunlight, maximum sunlight reaching the forest floor. So we expect species that are sun-loving to regenerate. So that is not really surprising in the ANR. And again, not surprising in the untreated site. When we go to the understory, the story is different. We have more of late successional species, obviously in the understory and then in the ANR site. But one key finding is that even in the understory, the seedling density that we counted in the understory of the untreated site, the bracken fern site, we still have more Macaranga kilimanjarica. One key thing is that, again, without this intervention, this is how the forest is going to look forever. Well, maybe not forever, that's overstatement, but that's going to take a long time to look more similar to the natural forest or attain the goal that we are trying to achieve. Now, to refer back to the overstory, while we have a lot of Macaranga kilimanjarica, that was an interesting observation that I saw in the forest. Now, these pictures are coming from the natural forest where there were closed canopies. In this particular areas, up. It could be one of the reasons why we have early successional species in the overstory within the natural forest, our reference point. So maybe this is something to take notice of. So I put in the geo reference for management if they want to follow this up, and I'm happy to share all the coordinate points of those areas. So I think I'm a little bit over time, but closing up, what we want to say is that ANR supports structural recovery, species richness, and diversity, according to this current research that we did in the forest. And then I would say that without intervention, untreated site remained less recovered structurally and then floristically. And then compositional convergence towards natural forest, it's really slow and may take decades to happen. So the forest after this treatment will basically look more like the natural forest. But this is a slow process. It's going to take some time to happen. But we have evidence that this is how the trajectory is going to look like in the future from this research. And then we'll also say that, assisted natural regeneration proves that this is a scalable restoration strategy in bracken-dominated montane forests. Thank you for your attention. I would like to say a big thank you to the organizers of this today's presentation. And then my boss, Mediatrice, I hope she's in the room. She really, really supported this work and ensured that everything was in place for me to work in the national park. And I really appreciate the support she gave us. I also like to appreciate the people I worked with, Jane Baptiste, and my friend from Banda, holding the Catlas over there. He's a great guy. I really appreciate his support. The Yale School of Environment, WCS, African Park, Protease, and the people that supported and funded this project, the 3Ks group in the US. Thank you for your audience, and I look forward to beautiful discussion with you all.
Wow, thank you very much. Yeah, from this research, we see that assisted natural regeneration works better in the post-fire event, in the post-fire forest management. Yeah, I thank you very much for the great presentation. I want to hear from the audience to see if any of them can have a question or comment. As usual, you can raise your hand so I give you time to speak, or you type your question in the chat room. The first press, I see Ja Emeru Tichu Mugambi, and he's in Yale University, maybe today. Yeah, I want to welcome him to speak to us.
Well, thank you so much. Thank you so much, Bena, and special thanks to Angelina for bringing these findings to the audience, to our attention. It's really amazing, and also was very important to hear such nice logical flow of asking some important ecological questions. So, but I want to ask you, like, if anyway, if you can answer the question whether you have really looked into the mechanistics behind the assisted natural regeneration, being like comparing regeneration or restoration strategy in the Afro-Montane regions. Did you look into different natural regeneration methods? Let's say a combination of 2 or 3 regeneration strategy, where you remove everything, or you remove the bigger or larger trees to release the smaller trees, or you just remove like the competition of the same age, or you remove all the wedding, what we say, wedding, it's like all everything. So what do you think is the best way, or a combination of 2 two or the other one?
Okay,
Thank you.
Eugene. I would say that the first response is that restoration strategy is not one size fits all. So you can't have one strategy that is the best, but you can have a strategy that is the best specific to a particular area, right? In our case, we didn't look into removing big trees and releasing. That would be like, forest management treatments to release specific species. But that didn't happen. In this specific study, this is more like monitoring, I would put it that way. In this particular study, fire had already occurred and bracken fern had already, colonized the area. And specific treatments, which has been tested already somewhere in 2003, and then they realized that 3 months was the optimum treatment scaling timeline. And so that treatment was applied. And then we went in to monitor and see how effective was that. So they had already tried 2, 3, and I think 5 months. And the 3 months was great. And now this monitoring or this research proves that this 3 months indeed is able to recover these forests that mimics the natural forest. But in terms of removing big trees will depend on the specific area or the context of the study. In this particular context, these trees were already gone. They were, consumed by fires. So there was no point in releasing, small seedlings or understory from big trees. And then again, the objective matters. If you want to do that, probably you might be interested in specific species for management purposes or for timber purposes. They do that a lot in the US. So you have a lot of black birch in black birch is a species that people don't really like in the US. They will be in a late stem exclusion phase. What people would do is that they would remove all those species and allow what they are interested in, which is in the ground cover, seedling stage, like oak or the maples, to take over. So basically, they would do release, clearing, cutting. But in this case, we did release. What was done was release weeding. They removed the bracken fern, which was kind of causing arrested succession because of the way, it grows and spreads. They spread through spores. So they are in the air wherever they find, and, they try to colonize. If it's entered rhizomes, they have this dieback system that falls. I think I measured this. It goes before you get to the mineral soil. When the leaves of the bracken fern die back onto the ground, before you get to the mineral soil, it will take you like 53 centimeters. We measured it on the average, 53 cents, which is very deep. And no seedling can survive in there, right? They'll just siphon the life out of it. So what people did was that they cleared this area, removed the bracken fern from the place and gathered it somewhere away from the plots that they have created and allowed the indigenous species that is in that particular place to survive. So that's what happened. So in summary, I would say that restoration strategy is not one size fits all. So I would not say one particular strategy is the best over the other. Rather, what I would say is that it depends on what the objectives are and then also the kind of system that we are looking at. Thank you.
Of course, you're welcome.
Yeah, thank you very much. Someone else, Bulomvu, Franklin.
Yeah, thank you so much, Vena. Thank you, Angelina, for the great presentation. And this is my interesting area because I'm finished my PhD at University of Rwanda. Mainly, I'm focusing in restoration by using indigenous tree species. That is very, very interesting for me. And
Yeah.
I would like to have any copy of your research, dear Angelina. I'm very, very pleased. So I would like to maybe to ask a suggestion or an orientation or like a question. Yeah, I know that the presence of bracken fern, Pteridium, aquilinum, is a sign of acidic soil. You know that most tropical soils are mainly acidic. And following your research, as you mentioned that you did not assess soil composition, yeah, this is understandable. And I think we can assume that this could influence regeneration. You know, there are some species which could be affected by the acidic of the soil, and this could be one of the reason. Maybe as you mentioned about Makaranga kilimanjarica, I know this is a widespread species in montane forest, and maybe, but I don't have any proud. Maybe maybe this could be influenced by presence of acidic soil. And again, I think that you mentioned again the other important point. You raised an important point to your graph when you mentioned that treatments, you support that treatment did not affect composition, but the presence of mature forests where birds and other animals could help to generate forest. That is a great point, and I agree with you because there are some presence of maternal plants influence the generation of species. So I would like to know if there is any hypothesis you thought about the presence of acidic soil which could influence also regeneration in both assisted and unassisted regeneration forestries. Thank you.
Thank you, Bulavu. I I really like your question and then submissions. I would say it's okay. Let me start from the report. So I'm happy to share the report with you, but I need permission. So I will look into it. And if I'm able to share, I will share with you. I'm glad that my boss is still here, mediatrice. Yeah. She's amazing. And so yeah, I will look into it and see if I'm able to do that. But then on the soils, because this study really didn't look into the soils, is the reason why we made sure that we collected the data from the same hill. So because we anticipated that differences in soils. So because if you collect data from hill and we collect data from a hill and we from another hill, what is going to happen is
Oh, sorry. F is the organization. Sorry. ESD organization. Can you hear me?
Hey.
Could you meet the FSD organization?
Hey. Your call has been forwarded to.
Eh?
Okay. Okay.
Yes, go.
Great. Thank you very much. For the acidic part of the study, we did not look into that. Our study didn't look at that scope. But it would be,
Where is the noise coming from?
right? Go ahead.
Yeah, sorry, Angelina, were you in?
Yeah, that's okay. It happens.
Yeah,
It happens.
Dan, we leave him from the meeting. Yeah, you can go on.
Okay, thank you very much. So as I was saying, we didn't really, make any hypothesis on the soil, which is going to be very interesting to look into. I would say that if it's going to be one recommendation, if anyone want to follow up this study, or if there's a chance to follow it up again, I would want to look into the soils, further into the soils, to see if the soils also has influence. But as it stands currently, we did not do that. Again, because we wanted to see, the pairing of these plots on the same hill to see what the influence looks like. Again, we anticipate that there could be differences in the soils, but not much because they are on the same piece of land, the same hill. So there might not be any differences. However, if we collected data from, the top of the hill and at the bottom of the hill, the soils are likely to be different, or even two different hills. The soils are likely to be different. I like to project that again. Okay. Right. So you can see the data collection were paired up on the same hill, as you can see in this map. So the treated side and untreated side are found in the same area. And then we compared it to a forest that's in a different place. But that's a great question. We would like to look further into that and see if the soil had any form of influence on this, which is very interesting. I like the submission, and I would put that into a recommendation. We actually thought about that, but we didn't do that as part of this work. Yeah.
Thank you so
Yes.
much.
Thank you for your question.
Yeah, thank you very
Okay.
much. We are approaching the end of our seminar today. I don't think you have the other questions or comments. But first of all, I want to thank you very much, Angelina Serwaa, for the wonderful presentation. And also, I am extending my gratitude to all participants here, especially Mediatrice Serwaa. I greatly thank her because she has become a partner of us, especially in CoEB seminar series, because she has been sending me contacts of people who can speak to us, especially MSc students who come to do research in Nyungwe National Park under the collaboration between WCS and the Yale School of Environment. Thank you very much, Ivana. Thank you very much, Angelina, and all the team here. It's all coming back. We'll meet again on the next occasion. And I'd like to remind you that our seminars mostly take place on Wednesday, 3 p.m. And if anyone has something to share, research, an idea of sharing, kindly reach out to us. Yeah, and again, Mediatrice Serwaa wrote in the chat room that the report will be shared to everyone who needs it. I know Bulomvu Franklin requested for it. If anyone
Yes.
wants it, I want his email so we can share it directly.
Okay. Let me share it with you now. Thank you.
Yeah. Also, other people who wants the report can share their emails in the chat room. I will collect them.
So, Vena,
Any
allow me
final
to
words?
send my greetings to Bana. Hello, Bana.
Hi everyone, and maybe I can say something about your question about removing old trees to allow regeneration. What is being done in Nyungwe is to remove exotic trees, but not native species, because it will be, it's against the law anyway. Yeah, that's what I would like to add on what Angelina saying. So, and when the report will be ready, we'll be happy to share with everyone. Thank you.
Thanks, Media. Good to hear from
Yeah,
you.
Thank you.
thank you
Thank
so
you
much.
so much.
And thank you very much for your presentation, Angelina. It was really
Thanks.
very interesting.
Yeah, it's very interesting, and this show me that knowledge which is not shared is almost useless. Yeah, but when the knowledge is shared, we share experience from one place to another. That's how we grow, actually. And
Yeah.
also, as in academia, it's another way of learning on how research is done, how graphs are done, and a way of making contacts. So we greatly value these kind of seminars we run every Wednesday. And the other people, when they run event, I make time to attend. Yeah.
Yeah.
Thank
Yeah.
you.
Yeah. Thank you very much. We stay in touch.
And
Thank
thank
you.
you.
Bye bye.
Bye.
Bye bye. Bye bye.
Bye
Bye.
bye. Bye.